Hi everybody. In this lecture we are going to be talking about transcription. So it's 7.2 in our textbook. It's the first step of gene expression. So when we were talking about the central dogma, this is the first part of it. So the copying of the gene onto messenger RNA from DNA, that happens in the nucleus. We talked about that before. The DNA can't leave the nucleus. It has to send a small copy of itself, just a copy of a gene, out into the cytoplasm. It's too dangerous for the DNA to go into the cytoplasm. So the first step of transcription is called initiation. RNA polymerase. We've heard of polymerase before when we were talking about DNA synthesis. This is RNA polymerase, so it's only making RNA. So the RNA polymerase binds on to a particular nucleated sequence. That's called the promoter. So it's the very beginning of the gene. At that point, it joins on and the gene itself unwinds and unzips. So you'll see this kind of bubble action happening in a diagram later on. The second part of transcription is elongation. So the messenger RNA is going to become longer once the polymerase joins on nucleotides. So it's the assembly of a single template strand of messenger RNA which is complementary to the DNA. Just remember that with DNA, adenine combines with thymine, but with RNA, the adenine will combine with uracil, cytosine with guanine. So adenine and uracil, because this is RNA. The third step is called termination. So termination, of course, means to stop. So the RNA polymerase moves along the DNA strand until it reaches a terminator sequence, a stop signal at the end of the gene. The new strand will be called the primary mRNA transcript. It's a copy of the DNA, not an exact copy, certainly not at this point. Three things have to happen to it before it can leave the nucleus through the nuclear pores. A 5' cap of guanine nucleoside triphosphate is added, so it gets a cap on top. A poly A tail, so off the backside it gets about 200 adenines. So about 200 adenines are added onto the 3' end, and then very, very importantly, introns. Introns, it says useless code. It's not perfectly useless. It's just not needed for making this particular polypeptide. So they're snipped out and exons are spliced together by spliceosomes. I love the name spliceosome. It's actually a group of enzymes. The final mRNA transcript leaves the nucleus through the nuclear pores. So let's take a look at the diagrams. So here we have the double helix that's in the nucleus, of course. The promoter is this yellow section right there. The RNA polymerase joins on to the promoter. The RNA polymerase causes the DNA to open up. You can see it's nice and wide and open, and only one of the strands will actually be copied, and it's not an exact copy, it's actually a complement. So this blob here moves along and makes a complementary copy of this strand of DNA using RNA. So it's the RNA copy. It's a complementary copy. And the amazing thing is it's actually identical to this strand because these two strands are complementary. So it is identical to this one, just using RNA instead. Stop sequence you can see in green. So when the stop sequence is reached, that's the terminator sequence. Termination happens. The RNA polymerase falls off. The transcript comes off. It's called the primary transcript. So up to this diagram, primary transcript, you can see it has something called exon, intron, exon, intron, exon. The exons we want, the introns we don't. So what happens next is a 5' cap of guanine is added. At the 3' end, the opposite end, about 200 adenines are joined on, so that's called the poly A tail. Both of these are for protection so that it doesn't get digested when it's in the cytoplasm by enzymes. And we have spliceosomes, excise introns, and joins the exons. So the introns are taken out, the useless information. The exons are spliced together because they're kind of like glued together. And now this is called the mRNA transcript. It can now leave the nucleus through the nuclear pores. Thanks.